So in today's lectures, we are going to discuss about two things. The first is functional anatomy of male reproductive system, and after that, I'll discuss about the spermatogenesis process. So let's start the first part of this discussion. So as we know that the reproductive systems is very vital for humans because it involves the reproductions. So for the reproduction purpose, obviously there must be the systems must be proper, and all the parts of that reproductive systems must work properly so that the reproductions will be effective. So here, since today's discussions, we will try to learn what are the most important structural part of male reproductive system and what is their functional features. After that, we will discuss about the detailed process of spermatogenesis. So, as it is mentioned here, the functional anatomy of male reproductive systems includes a group of organs and structures that works together to produce, store and transport sperm. So, here's not only the formations, storage and the transport of sperms is important. Along with this, reproductive systems or male reproductive systems is equally important for the secretions of male sex hormones, primarily the testosterone hormones, because we know the testosterone hormones, the most important male sex hormones that played an important role in reproductive process. So here's one by one, I'll try to explain the structural part and their functional features. So here's the very first, the most important part of male reproductive systems is the testicles. So if you see the testicles, it is basically located in the scrotums that is present outside the body. And here's the scrotum played an important role in male reproductive systems. How? Because if you see the characteristics of the scrotums, it maintains specific temperatures, right, which is around 2 to 3 degrees Celsius less than core body temperatures. So why it is important? It is important because these temperatures is important for reproductive purpose. Mainly the formations of sufficient number of sperm. So what is the most important functions of that testis? You can see the testicles. These testicles actually involves the formations of spermatozoa, which is also known as sperm. The formations of that spermatozoa or sperms is basically carried out by a specific physiological process. The name of that process is spermatogenesis, right? Here's these testicles or testes also involves the secretions of testosterone hormones, which is important for the male secondary sexual characteristics along with the male libido. So here's this is the most important structural part and functional features of testis. Now after that, the second part, which is also called the external part, is called epididymis. So what is the locations of that epididymis? You can see this is the epididymis, and this epididymis actually located the posterior surface of the testis, right? And so what is the most important functions of that epididymis? Yes, if you see the functional features of the epididymis, it mainly acts as a site of sperm maturations. Not only the synthesis of sperms is important for reproductive purpose, at the same time the maturations also important for reproductive purpose. So here epididymis acts as a site of maturations, along with it acts as a storage site for sperm. Now here's this epididymis also involves the motility of the sperm. Okay, so what we have seen here, this epididymis not only acts as a site of sperm maturations, it also acts as a storage site for sperm. Along with it also involves the motility of sperms, which is very essential for the fertilization purpose. So here's now what are the other parts? See here, Another important part is called this Vas Deferens, right? So what is the most important features of this Vas Deferens? Yes, if you see the structural features, it is a tube-like structures and it is muscular in nature. And these tube-like structures actually involves with the release of the mature sperms from the epididymis. Now you can see here, this is the Vas Deferens and this is the muscular tubes that travels from the scrotums into the pelvic cavity. So here you can see the Vas Deferens or ductus deferens mainly involved the transport of mature sperms into the ejaculatory duct. So here's up to this I have mentioned three different parts. The first one is the testis, then second one is the epididymis, and the third one is the Vas Deferens. All these parts are basically the external part. But here, now, next one is called seminal vesicles. So here, if you see the structural features of the seminal vesicles, it is paired in nature. So what is the most important features of the seminal vesicles is, it is just located behind the bladders. You can see this is the bladders and this is the seminal vesicles. And if you see the anatomy, you will find the paired seminal vesicles. And this seminal vesicles is very significant because it involves the formations of some fluid called seminal fluid, right? So what is the features of that seminal fluid? The features is, it has some chemical features. So what is the chemical features of that seminal fluid? It contains fructose. So this fructose is very important for male reproductive systems, mainly for the sperm cells. So why? How? Because this fructose acts as an important energy source for sperm cells. So it means the seminal vesicles which produce some seminal fluid is important in the developments or maturations of the sperms because this fructose which is present in seminal fluid acts as an important energy source. Along with this seminal fluid, it also contains some prostaglandins. So this is what is the important characteristic features of this seminal vesicles. Now see, the next important part is called the prostate gland. See here, after that seminal seminal vesicles, you will see this part is called the prostate. This is also just beneath the bladders. So what is the most important functions of this prostate gland? First, see, it is a single gland that is located just below the bladders and surrounding the urethra. This is the urethral canal, right? This is the urethral canal. So here's what is the functions of these prostate glands? It produces some prostatic fluid. If you see the characteristic features of that prostatic fluids, you will see it is alkaline in nature. So what is the role of that prostatic fluid? It helps neutralize the acidic environments of the female reproductive tract. So it means this prostatic fluid, which is alkaline in nature, is very important. That is useful during the sexual intercourse, right? Now, next part is called bulbourethral glands or Copper's glands. So what is the locations of this particular glands and how actually it looks like? So here it is also a small paired glands that is present beneath the prostate. So this is the prostate, and if you see the structures, and this is the prostate, and just beneath this is the bulbourethral glands, right? This is the bulbourethral glands. So what is the functional features of this gland? It secretes a pre-ejaculatory fluid, right, that lubricates the urethra and neutralizes traces of acidic urines. So this is the urethral canals. So these urethral canals is basically involves the two functions, release of urines along with the release of ejaculatory fluid. So here's these bulbourethral glands mainly secrete some pre-ejaculatory fluids that lubricates the urethra, as well as it also involves the neutralizations of traces of acidic urine. Now, next one is the prostate glands. See here, I have already mentioned that prostate glands, this prostatic fluids, which is alkaline in features, that this alkaline features prostatic fluid involves the neutralizations of acidic environments of the female reproductive tract. Next part is urethra, right? So this is the urethral canals or urethra. So as I mentioned, it performs two different functions, mainly the release of semen, along with it also involves with the release of urine. So here's you can see this runs through the penis and has three different structural parts. And if you see the functional features, it acts as a common phases for urine and semen. Now, see, this is the penis structures, and this most important functions and anatomic features of the penis is it is basically composed of three different segments, and these three different segments or penis structures mainly act as organs for sexual intercourse, as well as it also involves with the urination process. So, this also contains some erectile tissues that becomes engorged with the blood during erections. So, after this, we have seen that there are certain structural parts of male reproductive systems, and all these reproductive part or male reproductive structures are functionally important. Now, as I mentioned that male reproductive systems involve some structural parts. At the same time, male reproductive systems involves the formations or synthesis of the male sex cells. So here's the question is, what is the most important process that involves the productions of male sex cells? That is called spermatogenesis. Now we will see what is the exact process of spermatogenesis, or how an immature sperm cells is matured in this particular process. See here, this is a general flow of spermatogenesis. So what is the basic definitions of spermatogenesis? Obviously, it is a process of formations of sperm cells by specific process. So now let's see what are the most important events that is involved in this particular process. This spermatogenesis process is mainly involves some certain types of cell divisions, mainly mitotic cell divisions and meiotic cell divisions, both involved in this particular spermatogenesis events. So now say here the spermatogenesis or formations of sperm cells basically initiates from an immature spermatogonia, right? That spermatogonia is also known as the stem cells. It means this stem cells has the capacity to transform in any type of cells. But yes, the steps that involves this process is mainly it produces the sperm cells. Now question is how this two different cell division events involves with the formations of mature sperm cells or spermatozoa from spermatogonium. Here you can see, if you see the total number of genetic contents in the spermatogonia, it is diploid in nature. It means the total numbers of chromosomes in this spermatogonia is 2n or 46. Now, the whole spermatogenesis events is basically divided into 4 different stages. So, what are the most important stages? The stages are, the first one is the proliferation stage, second one is the growth stage, then the maturation stage and the transformation stage. So, at the very beginning, if you see the initial phase here in this spermatogonia, the total numbers of chromosomes is 2n, that is the cell is diploid in nature. But during the differentiation process or the differentiations of this spermatogonia basically occurs by mitotic cell divisions. So as we know, the mitotic cell divisions is basically the equational cell divisions, where there is no changes in the genetic content, right? So now you can see, due to the mitotic divisions of this spermatogonia, it produces a cells called primary spermatocyte. So if you see the chromosome numbers of these cells and spermatogonia, it is same. It means the events or mitotic events is the events is equational cell divisions, right? Hence, it produces a daughter cells with same chromosome number from its parent cells. So here parent cells is spermatogonia. Now after the formations of this primary spermatocytes, it undergoes the meiotic events. If you see the meiotic cell divisions mainly have two different stages, the meiotic 1 and meiotic 2. So we know that meiotic ones is called the reductional cell divisions, and the meiotic 2 is called the equational cell divisions. So here's the primary spermatocytes first enters into the meiotic ones. So in that meiotic ones, or by the process of meiotic ones, it again produces some daughter cells. So these daughter cells here is secondary spermatocytes. So what is the basic difference between these primary spermatocytes and secondary spermatocytes? They are chromosomal numbers, right? Here it was diploid numbers of chromosomes, but after the first meiotic cell divisions, it produced secondary spermatocytes, where the numbers of chromosomes is half of the primary spermatocytes. Now, if you see the most important part of this particular stage, the growth stage. If you see the growth stage of that particular primary spermatocytes, basically occurs in the prophase stage of meiotic 1, right? After that, the primary and secondary spermatocytes, here's mainly the secondary spermatocytes enters into the meiotic 2. So, as the second times of meiotic cell divisions takes place. Here's if you see the numbers of cells produced from secondary spermatocytes is 4. And at the same time, another important thing is, here's the number of chromosomes is haploid in numbers. At the same time, after the meiotic 2 divisions, it produces daughter cells where the numbers of chromosomes equals. It means the meiotic 2 here, though it gives the doubles numbers of cells from a single secondary spermatocytes, but if you see the chromosome numbers remain same. So now from that particular point, we could say here's this stage is basically indicates, though it is a meiotic 2, but it is an equational cell divisions, right? Because there is no changes in the chromosome numbers. So here's after the meiotic 2, the daughter cells produced are basically the spermatids, and that spermatids are immature sperms. That immature sperms undergoes some transformation events. So due to the transformations of that spermatids, it produces the mature spermatozoa, right? So the formations of that mature spermatozoa, spermatids, is called spermatogenesis, right? After the synthesis of that mature spermatozoa or mature sperms, the mature sperms will take place in the fertilization events. For the fertilizations, it must be motile, and that motility of this spermatozoas or mature sperms is known as the spermiations, which basically takes place in the fertilization events in the fallopian tube. So this is a very brief discussions about how this spermatogenesis events involves with the formations of mature spermatozoa from immature spermatogonia, or how this spermatozoa is produced, or haploid spermatozoa is produced from diploid spermatogonia. Now, this is just descriptive explanations of these particular events. Here you can see, as I mentioned, these particular spermatogenesis events involves the mitotic cell divisions, right? And that mitotic cell divisions actually occurs from spermatogonial stem cell, right? Here's, if you see the natures of the spermatogonial stem cells, these spermatogonial stem cells are of two types, and these are the type A spermatogonial stem cells and type B spermatogonial stem cells. And the most important part here is in the differentiation process of these spermatogonial stem cells. Here's spermatogonial B stem cells involves with the spermatogenesis events mainly. Now, here's, as I mentioned, the next cell division events here involves is meiotic 1, which gives secondary spermatocytes, and meiotic 2, which gives haploid spermatids, right? So it means this spermatogenesis events is very important because it produces the mature sperm cells, which is very, very essential for another physiological point of view, because once it produces the mature sperm cells, then only it is possible to fertilize the opposite sex cell, that is the egg cell, right? Now you can see from here, this is just a diagrammatic representations of a number of chromosomes in daughter cells. You can see this is 46 in primary spermatocyte also, it is 46, and after the meiotic divisions, first meiotic divisions, it gives daughter cells where the numbers of chromosome is half, and After meiotic II, it gives spermatids. It also contains the equal numbers of chromosomes, and finally, it gives a mature sperms. Now, question is, what is the times as spermatogenesis starts? Whether it starts at the very beginnings of the lives or it starts a specific period of lifespan. So, spermatogenesis actually starts at the pubertal phase. This is most important features of sperm or gametogenesis events. Another important features of gametogenesis or spermatogenesis is here the spermatogenesis or the formations of the sperm cells starts at puberty and it continues until death, right? This is the very important features of male reproductive systems. But if you see the female reproductive systems, the egg cells are produced. These egg cells are produced by oogenesis events, but the productions of that egg cells continues up to a certain period of times, means until the menopause, right? So here, this is what is the basic difference between the spermatogenesis or male sex cell productions and the female sex cell productions. Now see, here's what is the structural features of the mature sperm cells. This is a mature sperm cell, right? If you see the mature sperm cells, it is basically composed of 3 different segments. One part is the head piece, then the mid piece and the tail portions. And what is the structural features of these head piece? Yes, there is a specific portions called ectosome. So what is the features of that ectosomes? Ectosome contains some certain types of enzymes, and that enzymes is very important for the fertilization process. And that ectosomes basically releases some enzymes that is useful to penetrate the egg cell membrane, right? At the same time, the DNA content also presents into the head regions, because this head regions contains the nuclear portions, right? Other than this, the mid portions or mid piece is basically composed of mitochondria. So, as we know, the sperm cells are motile cells. For the motility of the sperm cells, it needs some energy, and that energy is produced by a mitochondria. So, this is how the mitochondria are located. And at the same time, the tail portions of flagellants is very important for the movements of motility purpose of that sperm cells, and it is important during the fertilization event. Now, you can see what is the most important purpose of spermatogenesis. Spermatogenesis produces mature male gametes, commonly called sperms, but more specifically known as spermatozoa, which are able to fertilize the counterpart female gametes, that is the oocyte. Another important purpose of this spermatogenesis is it involves the reproduction process. So, in this reproduction process, 2 different sex gametes takes place. Ultimately, they give a fertilized zygote. Now, what is the exact site of spermatogenesis? And what are the most important cell types that played an important role in spermatogenesis event? So, you can see the most important site is seminiferous tubules. So, why seminiferous tubules? Because these seminiferous tubules contain certain types of cells, mainly the Sertoli cells and Leydig cells. So, both the Sertoli cells and Leydig cells are equally important for the synthesis of certain types of hormone. Mainly the Leydig cells involved with the synthesis of male sex hormones, which is very essential for the spermatogenic event. Now, another important information says, in case of human spermatogenesis events, this process actually needs a certain period of time. So, what is the time required to complete a single cycle or to produce a single sperm cells, mature sperm cells? It is around 74 days is required to produce a single mature sperm cell. And another important information says, testes produce around 200 to 300 million mature spermatozoa daily. Though it produces these numbers of mature spermatozoa, but all are not functional. Some are functional. So, you can see, however, only about half or 100 millions of this becomes a viable sperm cells, right? So, though there is a synthesis and formations of around 300 million sperm cells, not all the sperm cells are viable. only around 100 million sperm cells are viable sperm cells, which can take part, participate in the fertilization events. Now, what are the most important influencing factors of the spermatogenesis? Yes, obviously there are certain factors. The most importantly, the environmental factors along with certain types of hormones and temperatures. These three are the most important factors that influence the spermatogenesis events, or that influence the formations as well as the maturations of spermatozoa or immature sperm cell. Now see, sometimes we can see the abnormality in the formations of the sperm cells or mature sperm cells. So what are the most important types of abnormality? One is called agiospermia. So what is the features of these agiospermia? There is absence of sperms in seminal fluid, right? Oligospermia, where the numbers of sperm cells is not sufficient. There is a decreased numbers of sperm cell productions. At the same time, though there is a decreased numbers of sperm cells found in the oligospermia, if you see the numbers of decreased numbers of sperms, if it is more than 20%, in that particular case, the persons may suffer with the infertility. It means this decreased numbers of sperm cells also affects the fertility process. Then necrospermia, which is denoted by the dead sperm cells present in the seminal fluid. Other than this, you can see sometimes the persons are unable to ejaculate sufficient amount of semen. So ejaculatory dysfunctions also as a part of abnormality of spermatogenesis. At the same time, some genetic factors also influence the spermatogenesis events. These genetic factors may be due to the mutations of certain types of genes or certain types of chromosomes, mainly the sex chromosomes, that affects the formations of either insufficient numbers of sperm cells, or it affects the it forms the abnormal types of sperm cell that ultimately alters the normal process of spermatogenesis event. So these are some important terminology that is related to the abnormality of spermatogenesis event. So here in today's discussions, I try to explain what are the most important structural and functional organs of male reproductive systems, and after that what are the different type of cell divisional events that takes part into the spermatogenesis process, and how this immature spermatogonia is converted into mature spermatozoa by the process of cell differentiations and both the mitotic and meiotic cell divisions. And after that, I have discussed what are the different types of abnormalities that is related with the spermatogenesis process. So this is what I have discussed in today's lectures.